Joseph. So yeah, thank you everyone for joining, and thank you for offering up your Tuesday night. It is Tuesday now. I always forget with COVID time what day it is. So let's go ahead and I'm going to share my screen here. I haven't done this as many times, so hopefully it works. Okay. Joseph, it looks like it's telling me that host is disabled again. So from sharing, so I think you have to make me either a host or I think right now I'm just a participant. This is live, everyone. So, oh, okay. So, all right. Pay no attention to the man behind the curtain. Okay, so hopefully this works now. Okay. Okay, that should work. All right, perfect. Okay, so here's my screen. Hopefully everyone can see this all right. Does everyone see that all right? I see your chat kind of coming in, so that's pretty good. That'll help me out a lot here. Okay. Oh, slideshow. Okay. All right, so thank you everyone, and thank you to CNPS for giving this opportunity for me to share my capstone project that I completed at a very new program at UC Irvine. This program was created out of a need for people to have a specified advanced education in conservation and restoration science to actually learn how to do this in a real practical way. So thank you for letting me share what I spent the last couple of years researching how to do. So this method is it's as Joseph mentioned, a tool that can be added to the tool belt for conservationists to be able to conserve species. So we do a lot with seed banking, and also traditional propagation like we do here at Tree of Life with seeds and cuttings. But this is another way to conserve the species with utilizing the lab environment to be able to grow plants. It's also a method for production that allows for rapid production, which is what we're going to go into about how this is actually a beneficial tool that can be used towards the poaching effort when there's not a lot of plants available. So without further ado, let's get into the presentation. So the main question, or a good place to start, what are Dudleyas? So many people may know them as commonly as the name live forevers, but genus is the botanical, or the Dudleya is the genus for the plant, or the botanic name, of which there are many different species, but commonly they are called live forevers. They are a group of succulent plants in the Crassulaceae family. You may know that family for things like echeverias, which are the things like hen and chicks, or even things like your jade plant. So Dudleyas are relatives of these plants. As the Crassulaceae name also implies, that they have this technique of photosynthesis that allows them to survive in harsh environments and effectively do their photosynthesis and still store that energy and use that energy in a method called Crassulacean acid metabolism, but a lot of people just call them CAM plants, which the Dudleya are one of those. It is a succulent, so they do have succulent parts, which they're able to store their water and nutrients and other types of things, primarily water, in these parts, so that they're able to handle drought conditions. And you typically see them in two different forms. In a rosette form, which you see here on the top right here. You may recognize this plant, especially right now, that's in flower, putting up these big flower stalks. This one here is called chalk live forever, or Dudleya pulverulenta. I did have to practice how to say that one when I first learned it, but I think I got it down by now. Where it has these finger-like forms, which you see down here with Dudleya attenuata, or Dudleya edulis is another one. viscida. A lot of these have these finger-like forms, which you can almost see like this little rosette inside there too. So, which is near the growing point of the plant. Not letting me change my screen. Hold on, there we go. Okay. So there are approximately 45 species of Dudleya. That kind of depends on who you talk to. There's a lot of debate over different species and different subspecies, varieties, things like that. But primarily, you could say there's approximately 45 species of Dudleya. They occur throughout Southern Oregon, all the way down into Baja, California, and Mexico. And they primarily occur around coastal bluffs and slopes in these nice environments that have this coastal influence. But you do see species all the way in, that go into our canyon areas, on coastal on canyons and more inland, and even into desert areas out into even our Sonoran Desert and into areas into Arizona as well. One example up in the more northern part of their distribution is Dudleya farinosa. This occurs in the coastal sea bluff area, and we'll get into a little bit more of this species later on why this is kind of an interesting species for Dudleya itself, but very attractive, gets this really incredible red kind of color out into areas once it gets kind of a little stressed into this later spring and into summertime. It's putting on these gorgeous flowers. Dudleya cymosa is another one you see more inland in the canyon areas, up in the mountain areas as well. There's many rarer subspecies of this as well, and if there's any desiring botanists out there that want to really try to figure out the different subspecies of cymosa, it's a challenge. So good on you for trying that. As well as I mentioned earlier, Dudleya pulverulenta. It's a little bit more widely distributed. You see these a lot in the foothills and on coast or on canyon sides and in different slopes. But this one you may see more often, especially right now. It kind of grabs your attention in different areas. It occurs on the coast as well, also a little bit more inland. This one, speaking to the San Diego chapter, maybe a little bit more familiar with this one, but this is a bit of a rare species that occurs down into San Diego County. It's a subspecies actually of a tenuinata, which is a little bit more common, but the subspecies or charismatic occurs down into near the border areas of down in southern San Diego County and into northern Baja as well. You can see it has that finger-like form as well, as I mentioned earlier. And then out onto our Channel Islands. The Channel Islands have a special place in my heart, and so as do Dudleya. And so it's two of my favorite worlds collide, that there are many Dudleya species as well. This one occurs on Catalina Island, or the Santa Catalina Island Live Forever, which is Dudleya virens hassei. You may be a little bit more familiar with this one. This one is becoming a lot more common in the horticultural trade, which I'm very excited to see. But it is a rare species. And so hopefully that continues. The trend of this plant becomes a little bit, that keeps going, because I would love to see more of these out in the environment and in the trade. Out in Santa Barbara Island as well is Dudleya traskiae, or the Santa Barbara Island Live Forever. This one is pretty rare. It's also becoming a bit more common in the horticultural trade, although it is a state and federally threatened species. So so hopefully that this propagation of this plant continues, and that that it does not go extinct. But as I mentioned earlier, that a lot of these plants are rare. There's about 9 taxa of federally listed endangered or threatened from becoming extinct. And rarity in itself is kind of a common trait with Dudleya. Again, so if there's any aspiring researchers out there that want to look into why exactly that is, some people point that towards either habitat specificity or a lot of different options or factors. So it's a growing area of research, and I encourage anybody out there to take on that work. as I mentioned, there's about 45 different species of Dudleya, and 40 of those taxa, so that includes things like subspecies and varieties, are included on the CNPS list of rare and endangered plants. You may see like this one of my favorites. I grew up in Laguna Beach, so this one is by far my favorite plant, but the Dudleya stolonifera, or the Laguna Beach live forever, that occurs in only a few canyons in Laguna Beach. But as you can see, it's state and federally listed as threatened, but has this ranking system of endangered plants that maybe a lot of people here in the CNPS know, with the 1B1, which is one of the highest ratings you can get for rare plants. So even though it's not federally listed, it still can be considered listing as threatened by the CNPS, and good on them for doing that. Another species is Dudleya Verityi. This occurs more in the Ventura County and up in parts of the mountains up and around there. It is federally listed as threatened. It is a 1B1. And unfortunately, this plant has gone through some real challenges lately. It grows with a lot of this association with lichen, as you see here in this photo. And that's something that people up in at the Santa Barbara Botanic Garden are doing a lot of research now on how to grow this plant a little bit more and help conserve it and understand that relationship with the lichen itself. But in 2013, this species was severely impacted by the Springs fire up in Ventura County, where 95% of the habitat and the Dudleya there in that habitat were destroyed in this fire. It was a very intense fire, as we're seeing more and more often in California. But actually speaking to some people, that this is probably a conservative estimate. It's probably more closer to 99%. But again, that's who you talk to. And so, but it doesn't seem to be, either way, it's 95, 99. It's a horrible thing that has happened for this species. And it's not a species, and Dudleya in general is not really a species that's known of whether or not it's really a fire adapted plant. We're gonna have to see the case with that, but it doesn't appear to be the case. So, but overall, the most important, or the most prevalent just threat to Dudleya in general is habitat loss. So like I mentioned earlier, I'm from Laguna Beach, and this is an area here where I grew up in Laguna, and you can see just these plants, usually Dudleya in general, occur in the coastal area near the ocean and near the beaches. And this is prime real estate for Dudleya, but it's also prime real estate for development. And so these species, it's the #1 type of thing that is threatening the genus overall. However, a new threat has surprisingly hit the plants. And so maybe a lot of people who are tuning into this presentation have heard and want to learn a little bit more about. But recently, back in 2018, the California Department of Fish and Wildlife received an anonymous tip for what they anticipated was possibly abalone poaching up in Northern California, and just the suspicious behavior and what was going on. And what they found was actually a little bit more surprising. What they found was that people were poaching plants of Dudleya, primarily these succulents, in very large quantities, as you can see, especially here on the right-hand side. And so there, at some estimates, I think it was even close to tens of thousands at a time. But either way, this could do really severe damage to a population. And this occurred primarily on Dudleya farinosa, which occurs, and it's not really a rare species as far as Dudleya goes, but if you're poaching at this level, you could easily wipe out entire small populations in different distributions. So you can see how this is just severely impacting the gene pool of the genus. And so they found that this was primarily to ship plants back to a growing market in Korea, but also China as well, but primarily Korea. And as a rare plant, it's not commercially available, and this real chalk-like nature that you don't usually see on many other plants is very, very attractive. But it's also kind of it's a really bad situation. Our climate here is not the same as it is over there. So it's not real likely that these plants will do very well over there unless real care is taken. But it's a very, very sad situation and a very serious threat. Usually when you hear of poaching, you think about, you know, rhino horns and elephant tusks and things like that. But unfortunately, there's not a lot of effort and initially thought on things like plants themselves that are poached. They are desirable, and they need our help. So it's pretty bad. And it's likely that farinosa was targeted because this is kind of the prime plant that a lot of these markets are looking for. It's a rare species that occurs only on Cedros Island down in Baja, Mexico. It's called Dudleya pachyphytum, and you can see it has a very unique shape and color and everything like that. And this was poached, and some extreme effort went into wild harvesting this population off the island. And so they have nowhere else to go. They are from that island, and they only occur on that island, and they were hit pretty bad. And so I think what happened was that they started to tighten up the regulations on it, or at least their protection on this genus, and that made them kind of go northern and up into a kind of similar looking ish plant, the Dudleya farinosa. But so I'll get in a little bit more about these particular species, but yeah, it's a really bad and heartbreaking situation for any Dudleya enthusiast out there. As I mentioned earlier, that Dudleya verityi is really bad. Maybe 99% of that population has been possibly destroyed after the spring's fire. But unfortunately, in discussion with the U.S. Fish and Wildlife Service, they have confirmed poaching on that 1%. Who did it? It's not exactly sure, but they did find evidence of that, which is a very scary factor if there's only 1% of that population left. So there's things you can do. The CalTIP, like I said, that what started this entire thing of sending the California Department of Fish and Wildlife out there to do investigate potential poaching. There is an anonymous hotline. So either you can call this number or text. You know, if you're not in a situation where you can make an audio phone call, you can text to TIP 411 and report these types of findings, and they'll look into it. And I encourage you to do this for not just Dudleya, but any wild harvesting, poaching. This is the area that is growing, as that you may see a lot more wild harvesting happening, and these departments and agencies need your help, and to be citizens to help protect these plants. So. Okay, so where this all kind of comes from and why does poaching happen in the first place? It comes down to a simple principle of economics. And so especially since a lot of these Dudleyas are rare and they are becoming more popular, and there's a large demand of them, but there's a very low supply of these plants. And so because of that, these people that are doing the poaching, they are willing to spend the resources to return on their investment and get large high dollar demand for those plants. So if there's not a lot of plants on the market available, and there's a huge demand, then that's where the profits come from. And so that's where things that we can do is reverse that. And so what my project looked for was to utilize plant tissue culture. But just in general, just growing these plants is a method to reverse that and making these plants more available. So San Diego chapter, or any chapter that's of CNPS that's listening in on this, you're doing a great service by allowing these plants available at things that like your plant sales. And so not only is it helping you supply these plants, but you're also supplying a ethically produced plant. It's plants that are not poached out of the wild, and it makes it possible for people who may not think of it as an ethical thing, but they're able to ethically purchase a plant and be able to buy that plant. Where if there was a huge supply of these plants, then that cost factor drops, and that's economics. So if we're able to have a lot of these plants on the market, then that demand is deflated, and they won't be able to get these high dollar amounts. I don't want to say how much they get or have been able to get, because that actually can do a reverse type of incentive, where then people then know how much you can almost get for these plants. And so I won't answer that. If anybody has that in the question section, it's best not to, because after these press releases and things were going out about the Dudleya and how much, you know, that they were making on these black markets or markets out in other countries, it started to then have poachers go out and start doing this here locally, so in the US. So but the point is, you make these plants available and then flood the market, and hopefully we can deflate that poaching incentive. And that's a method that a lot of people have coined as conservation through cultivation. And so by growing these plants and learning the best ways to do so, we can ensure that these species survive, but also deflate these poaching incentives and make sure that these plants are there for everyone in the future. So Dudleya is actually relatively easy to grow from seed. I mean, it depends on a lot of different factors, but they do luckily put out a good amount of seed that do pretty well in germination if the seeds are viable. But there are challenges in propagation. And so, like I said, traditionally they're done by seed, although cuttings can do okay and set new roots from different cuttings of plants. But overall, if there's a lot of rare plants, which many of these desirable species are, then the resources for available seeds is very limited. And so these are either used for then be stored in seed banks, but may not be available for production and for people to then purchase and have these plants in their garden. Or for even restoration if it's really low amount of seeds available. But also, if anyone has tried to grow Dudleya with any other species around, Dudleya is notorious for hybridizing, and so and a lot of these species have very similar traits, so it's hard to even know if you have a hybrid. But this can occur and can be a challenge if you're trying to conserve a particular species. Hybridization also occurs in the wild. There are a lot of species overlap where you may have a more commonly distributed species in and amongst around the more rare species. And so that's a challenge for conservationists as well. So hybridization is a big problem with Dudleya to ensure that you have the exact species you're trying to conserve. For example, this is, like I said, one of my favorite species, and recently we've been we were trying to grow it here from some seed sources that were given to us ethically, of course, and from a private collection, and grew it all the way up, and here it is. I took this one home after growing it here at the nursery. And I'm like, wow, this Dudleya stolonifera is getting larger and larger. This is the biggest Dudleya I have. Maybe it's my fertilizer and everything, and it just continued to grow. As you see here on the right, it is not nearly as big as this. And the stalks, largest thing ever. And then realize, oh yeah, this was grown, you wouldn't think, you know, directly next to a plant, but this is likely a Dudleya bretonii and stolonifera hybrid. And so you imagine if you tried to grow this for restoration and you get to this point until you realize that you had a hybrid that you were trying to conserve, that can be a challenge and not the right thing to do. Very attractive, and it's probably a great horticultural plant, but you can see how easy these plants can hybridize. So one potential tool to conserve these plants is a method called plant tissue culture. It's called by a few different names, and that's usually by the different techniques that are used, but it's sometimes called micropropagation. But I like to use the term in vitro propagation, but this is it's not really a common tool used in production. It's becoming more common and more available to the home propagator and somebody who wants to give this a try. Usually it was kind of limited just to the universities, but now there's different kits and things you can buy and websites that are making this a lot easier for the home grower. And as more people are doing it, more information is out there for people to do that themselves. This is a method of, so what is it? It's a method of in vitro culture. So this is growing plants in the lab. Simply said, this is growing plants in test tubes. And that's actually where the term in vitro comes from. So in glass is the meaning, in vitro. And what it is it's growing plants in a very clean and sterile environment. You remove all the microbes and everything like that. And you grow these plants instead of soil, you're growing them on a nutrient agar. And so what that is, it's basically like a seaweed extract, much like Jell-O. But the ingredients in that, it does have sugar, much like Jell-O. And it has all the nutrients and things for that plant to grow. And so you also are providing ambient light temperature, or sufficient light and a nice growing temperature in a very clean room and setting in a laboratory. But you can also include hormones, natural occurring plant hormones in different concentrations in this nutrient agar to actually be in the driver's seat. If you, there's things called cytokinins that produce new plant parts or shoots. It's kind of called a shooting hormone. And then there's also rooting hormones or auxins. And you can actually put these in the nutrient agar at different concentrations to kind of be in the driver's seat of what exactly you want the plant to do. These, like I said, they're naturally occurring hormones, but there are synthetic chemicals out there from those naturally occurring plants that do the same signaling as well and same response. I learned this method and was highly inspired by this is Nellie Sugii at the University of Hawaii at the Lion Arboretum. That's where I did my undergrad at the University of Hawaii on Hilo on a separate island. But their work there was inspirational to me and kind of one of its kind. That highly uses this technology to conserve the species out on Hawaii. If you know, Hawaii has the highest rate of endemism, or plants that only occur in Hawaii, and also a very high level of endangered species as well. And so a lot of those pollinators are either extinct, and or there's many challenges in these species in the island currently. So what they did out at the Lion Arboretum is they use plant tissue culture because a lot of these plants may be very difficult to grow. Like I said, their pollinators might be extinct, or just they do very low or do very poor from seed as well. And also cuttings. Maybe these traditional methods of seed and cutting do not work for these plants, and these plants are in peril. Many of these plants, I think, they focused on plants that had less than 10 individuals in the wild. I think many, then they were able to grow into a lot, to just real priority species that are in demand for conservation. But they're able to grow these plants using plant tissue culture, but also provide this safe haven, much like seed banking, to continue propagation for these plants and ensure that these plants are not lost in the wild, and the genetics of these plants are not lost forever. And so Nellie has been doing an incredible work over there, and her staff. And so my inspiration after meeting with them was to bring this to California. And so it just happened that right when I was starting to do my master's capstone, that the poaching hit the news. And so I thought, okay, tissue culture is a perfect way to store the genetics like this, but it's also an incredible way to produce plants that the agricultural businesses and firms are doing with a lot of our food crops. So there's something that this tool can be a very effective tool for production as well, and I'll get into that in a second, about how you can actually deflate the market and the poaching incentive. So what is plant tissue? The different stages. So you take an initial plant here, you clean it in a chlorine solution, which is typically chlorine bleach or your commercial bleach product. And then you establish that onto that nutrient agar that I said, and you want to make sure that you clean the plant enough to remove any fungus or bacteria or any other microbial type things that could then easily take over this growing environment in vitro. And you're basically supplying all the nutrient and everything to do that'll make those fungus proliferate, but you want to make sure you're also not killing off the plant and providing too much damage to the plant itself. And so the only thing growing in that test tube is the plant itself. Like I said, you can add hormones into the media itself, and you can signal for new plants to grow. And this is cloning, and it happens as you see a new shoot come out, or if anyone's taken a cutting, and that is a clone, genetically identical to the initial source plant. And then after multiplication, you can then divide those plants out and then either include the rooting hormone and start to initiate roots to set. And then you have you have then divided and produced brand new plants from that clone or from that initial source plant. And that plant is a clone of the original plant. You then can much like you would take a plant from a greenhouse out into the environment, you acclimate them from these different environments. You would do the same thing from the test tubes itself, this high humidity, high perfect conditions, basically, out into the greenhouse and then out into the wild itself. So those are the stages of plant propagation. But to explain how this can be used as a production tool, and what the goal of this project was to grow a lot of plants to flood the market and deflate that incentive and that price of these plants. It's very similar to the tribbles. So I think Dr. Spock does a great job of explaining this. So hopefully the sound works.
There must be thousands of them.
Hundreds of thousands. 1,771,561. That's assuming one tribble, multiplying with an average litter of 10, producing a new generation every 12 hours over a period of 3 days.
And that's assuming that they got here 3 days ago.
And allowing for the amount of grain consumed and the volume of the storage compartment.
Kirk, you should have known. You are responsible for turning the development project into a total disaster. There must be thousands of them. There must be.
I can go to the next slide. So thank you, Dr. Spock. So so that's actually pretty spot on to how you can do how plant tissue culture works. So this is called exponential cloning or exponential numbers, where if you had one plant and you were able to then multiply that one plant by 10, that it usually takes maybe a couple months or so to do so. You can then divide those 10 plants into 10 different plants. So these are one plant cloned different 10 at a factor of 10. And so these are basically 10 cuttings that came off of one plant that you initiated with that hormone. Those 10 plants, then on that same hormone stimulation, you then have 10 more plants. And so now you have 100 individuals. And this happens, this could exponentially grow your numbers relatively fast by taking cuttings of cuttings or clones of clones, and they are multiplying like tribbles. So as you can see, very quickly, you can increase these numbers. Now this is optimum. Of course, there may be factors that may diminish this exact outcome, but theoretically, you could have within 6 transfers up to a million plants. And so that's what's being used a lot with some of the food crops that we have and other things like that to produce these numbers. A lot of the ornamentals plants there as well. These companies may have done this research and putting out a lot of plants using this tool. And so this is why I think it fits really well to be able to flood the market to decrease the poaching effort or the poaching incentive to provide these plants to the market. So that's what the goal of this project is. I needed to figure out the initial stages and be able to figure out how to bring these plants into in vitro culture. But the overall goal is to develop a tissue culture protocol specific to Dudleya to best grow these plants and deflate that poaching incentive, but also provide an additional tool for conservationists and to be able to store plants and store living tissues of growing plants in the lab in a safe environment, much in addition to the seed banking as well, so that we know that you don't put all your eggs in one basket to be able to conserve a plant itself. So when I began this project, I met with the Dudleya guru, Steve McCabe, the director of research up at UC Santa Cruz. He's done a lot for this genus, and when I first kind of talked to him, it was kind of funny. He was actually kind of waiting for someone to come by with this technology who had an experience with it, and he was very excited, and I was very excited to meet him as well, as someone who has an affinity for this genus. And we sat down up in Santa Cruz and came up with a punch list of species that we want to focus this technology on. And so it was hard to really come up with a list. Again, this is just part of my master's, and so I only have so much time to really complete this. But what we focused on were species that were really rare, but also targeted by poachers. And so something that like Dudleya farinosa, it's technically not rare considering compared to many other genuses or species or subspecies or varieties of Dudleya. But we want to include it because we want to deflate the poaching incentive. And so we came up with these plants where some that were needing help and also will increase the amount of available plants out on the market, but also include these plants into conservation realms as well. And so the scope of my master's project was just to get to these initial stages with the time I had. And so what I'm really looking at is the sterilizing and getting these plants established into in vitro culture, because that's where the most effective method for conservation is as well, and also is with the time I currently had for this master's project, but I'm going to continue this since I have graduated as well. And why I say it's the most important stage is because once these are clean and growing healthy in vitro, then they can be used for different avenues. So either it's for germplasm storage or to be able to store the genetics of that plant, much like seed banking, as I mentioned earlier. So these plants can be in culture, and you can decrease the different concentration of nutrients and things like that, and slow the growth itself, and store these plants cleanly in a lab setting. And so to make sure that genetics isn't lost. Tissue culture and in vitro, when you need the plants clean like this, is actually the initial stage towards cryopreservation. And so that's actually cold storage and freezing the plants basically in suspended animation. And you basically could then conserve the genetics of that plant for indefinitely. But as long as those plants are maintained at that temperature, and then you can bring them out and hopefully they start to live. And there's a lot of research being done at the Huntington Library with this as well. Or once they're in vitro, then you could go into the production phase. And so that's where you make these plants then available for horticulture, the horticultural market, for botanic gardens, for the home gardener, but as well for restoration, although these are clones. So that's up to the agencies themselves to make that call of how many clones you want to put out of a genetic source. But at least that option is on the table. So my number one research question was to the growth between in vitro of seeds, so how they grow from seed as the initial seed source, or in the greenhouse itself. So traditional versus this this non-traditional method, and see what the differences are. Is there a benefit to starting seeds in vitro that maybe we didn't know? And maybe that's the way to go versus taking cuttings and putting them into culture. But just overall, how does seed grow differently than traditionally in the in vitro environment? And so what I did is next time, although I love Dudleya, I had to use a microscope to see the seed itself. So it's a little challenging on my eyes to be able to separate and clean out a seed that looked viable. And then I sowed those seeds on different media types and different concentrations of that nutrient in vitro. And then also sowed the exact same number of seeds in the greenhouse. And then was able to compare the things like the germination rate, the survival over time, and the size and growth, and then note any abnormalities or any things that may have occurred. And what I found was that there wasn't necessarily a general trend or difference in germination, but was very clear was that every seed that germinated in vitro survived. This was over about a 5 month period. And but those that that did germinate in the greenhouse environment had many mortality. And so that's where you see here is where some of them were very, very low. So for something like Pachyphytum, didn't have very great germination ex vitro, which means in the greenhouse or outside the glass or outside the environment. And you see just almost half or more of mortality. So so overall, it's the in vitro helps in the survivability of the seed itself. And then as far as growth, there was no significant difference between the plants and under the different nutrient concentrations in vitro. But surprise, surprise, those grown out in the environment or in the greenhouse grew much, much faster. So these plants actually maintained a more juvenile state in the laboratory setting, which if you're looking to conserve just the genetics of the plant, that's a great thing. These plants are not spending all the time and resources into then putting out more seed, and instead are just chilling. They're just maintaining in the lab and nice, healthy environment. And also, from a management perspective, the greenhouse environment, I had to be watering these, and I had to be maintaining these and looking at insects and things like that, which I did have. I did have some moths, actually like caterpillars in some of these young plants, which you don't see in the lab. These plants are completely clean and supplied everything they need for nice, healthy growth. And so that's the benefit of using this. The second method is that, okay, so if you have a plant, how do you then bring a existing plant or a cutting into the in vitro environment? So that's where I'm going to look at how to clean these plants effectively without killing the plant or damaging the tissue. And so that takes some research. And so what I did is I used a test subject, Dudleya burtonii, which is a little bit more common in the horticultural trade than some of the other species that might be becoming more popular. But this was a good test subject because some of the rare species I only had, let's say 10 plants or less, and so or 10 cuttings even. And so I wanted to be able to figure this out before applying it to some of the rarer species. And so I had to develop an effective protocol. And so I took this plant and I compared two different cleaning solutions, the commercial bleach at different concentrations, and also a less common, and I think deserves a lot more focus, is NADCC. It's a different type of chlorine solution, but it is apparently less harmful to plants. And so I wanted to test that here and see how much plant tissue damage it does, and what would be an appropriate protocol for using either or, and compare these two treatments. And then I established them and tried to see on the same concentration of media and then note if there was any contamination and the degree of tissue damage or death of the plant itself. And you want to find that the treatment that has the lowest contamination and the lowest amount of damage. And so these are the different treatments I have, just 2 different concentrations of bleach, and put them at different exposure times, and either 10 minutes, 20, and 30. And as you would expect, those that were exposed to higher concentrations and over a higher time damaged the tissue, and I killed off plants. But you would have more chances for the microbes, the fungus and bacteria, to survive at lower concentrations. So it's a trade-off and where you got to find your success. And then for the NADCC, I had a comparable treatment to that was most effective by bleach, and tested that. And I saw, but I also added a different treatment as well, where I would then, after doing the cleaning, I would then remove some leaves and give it another quick dip, as I called it, to then be able to clean these plants even further. And I compared it if that was an effective method as well. And what I found was pretty good. So this is where the research comes in. So the tissue damage, or necrosis, or necrotic tissue, I measured that visually and saw that higher concentrations over higher time was you would see more occurrences of necrosis, where this one method I was able to select with NADCC, which you see in blue, was much more effective and minimized that tissue damage. And same as contamination, that method was also provided the cleanest amount of plants and the less occurrence of that contamination. And you can see this kind of dips up as you get a little higher, because you're not only are you killing off the microbes and lessening that amount, but you're also killing the plant. So that's not an effective treatment. So the NADCC at this treatment was the best one overall. So that's part of that protocol development. So once I had an effective treatment selected and identified, I then felt comfortable to then apply those to my rare cuttings, like this plant I had here of Dudleya Rarity. This is all I had to work on from some of the that was made available to me, and was able to then apply that treatment. And so from those plants we selected, I had exceptional results, actually better than what was on Dudleya Bretonii, which awesome. And in fact, in some cases, I had 0 contamination and very low necrosis or tissue damage overall. And actually, so things like Dudleya noma, the munchkin Dudleya, my favorite, the Dudleya stolonifera, verityi, and even hassei, I had 0 contamination. And so we got every cutting in there clean and healthy and growing. And some of the other species like pachyphytum and farinosa, we still got way better success than Dudleya Bretonii itself. But these are acceptable levels. And so I'm very happy with that. So now that was the scope of my master's, and was able to develop those protocol for the initial steps, but this will continue. And so this is what I'm working on now, and into the future. So now I'm going to work on, okay, how do I multiply these plants and then get these going and do that exponential growth, and how to root them effectively as well, and then acclimatize them out into the ex vitro environment in the greenhouse and out into the growing plants, and then hopefully in your yards, and make these available for people in the market. And hopefully we can just flood the market and bring that price point way down, and so that there's no longer this poaching happening, and there's no incentive if we can make these plants pretty cheap and ubiquitous. So that's kind of the scope of my project. And again, so just to recap, as far as seeds, in vitro culture didn't necessarily enhance. I couldn't really prove that statistically of whether it enhanced the germination rate. It did for some species, but I would need kind of a few more accessions and different collections to really prove that. But it did, the survival of those plants, every plant that germinated survived. And so that's really good for conservation to know that. especially if you have very low populations of plants, that you're able to increase the amount of plants within that population. So this can be a very effective tool by trying to grow plants in vitro. And then future directions, like I said, I was able to identify an effective cleaning protocol for these species, but I need to complete that protocol. And so once we can do that, then these plants could be then out in production, but also for conservation. But at least we know how to grow these plants and put them into sterile culture and in the in vitro environment, and hopefully that we do not lose these plants in the future if anything were to happen. So thank you for listening. I am available. Here is my email address if anybody has questions and we don't get to your question in the comment section. We will select a couple, but we want to, but I'm available. If anybody knows me, I work here at Tree of Life Nursery up in San Juan Capistrano in southern Orange County. And so come pay us a visit, and I'll be happy to chat Dudleya anytime with you, or just native plants in general. So thank you all very much, and I'm going to give it back to Joseph here. Joseph.
Here we come. Well, thank you so much, Kevin. That was just beautifully presented and Trekkie friendly to boot. We had a lot of Star Trek comments in the sections, including the reminder from Leon to live long and prosper, and
yes,
Brian quipped live forever and prosper.
Oh, good. My undergrad at the University of Hawaii at Hilo, my mascot was the Vulcan. So you know, so again, live long and prosper.
Excellent.
Pretty apropos.
Before we begin with some questions, I want to quickly mention that CNPS San Diego has a new online store selling at this point mostly California native plant seeds, along with some clothing merchandise. And I just counted 6 species of Dudleya seeds on the site. Okay. And through the end of this week, we're offering free shipping on all orders of $20 or more. I'll put the magic code for that in the chat feature on Zoom and in the comments on Facebook for those watching there. Also, everyone who registered on Zoom are gonna see that in a follow-up email tomorrow morning. So let's start with, there were a lot of questions, and I'm picking maybe the things that had broadest relevance to the audience. So Amanda asked very early on, did I miss discussion of when and why these became Dudleya instead of Echeveria?
Well, they're related plants, so they both are separate genuses. They look very similar, but yeah, Echeveria is a completely different genus of plants. Those don't occur in our region. Those are more down in, I believe, Mexico and even further into Central America. But yeah, they are separate plants. One thing that, interestingly, propagation-wise, people think that you could possibly grow Dudleya by grabbing, removing a plant or a leaf and growing that plant, which you see new plants sprouting. That's very common with Echeveria. It does not occur with Dudleya. And so that's one of the reasons that having another tool like this can be effective with plant tissue culture for cuttings.
Thank you. Mark asks if you could give a little more detail about the specific setting where some of these are found. For example, like bare rocky space, grassy space, near shrubs, etc. And I will say that Robert writes, and this might be a rough but good summation, that in San Diego, Dudleya are found mostly on slopes and canyons within about 5 to 10 miles from the coast. But I know that they extend further, like into desert margin, and then some are very, very like right on the coast. So can you just—
That's correct
about that?
Yeah, so that all comes down to the species itself. So they've gone through speciation based or that's something that could be researched as well, but based on different climate types. And so putting on different generalities to the species itself can be a little more difficult. But yes, they do occur in different regions, but primarily a good amount of the species occur in the coastal regions. You do see these seeds and seedlings grow, as someone mentioned, in and around other plants. So there are plants that do give that what's called a nurse culture for these Dudleyas, and even a little bit protection, maybe from herbivore or different herbivores and things that may be wanting a nice succulent treat. But at least it allows these juvenile plants to grow up into larger plants. You typically will see that more inland, and a lot of the more plants you see growing out into even desert regions, those will have a lot more chalk on them. That giant chalk live forever, occurs on coastal areas, but also in real exposed areas on hillsides. And so that chalk can act like a sunscreen. But yeah, there's many different types of Dudleya, and so they have different adaptations based on where they occur. And so, yeah, so it all comes down to speciation.
Sonny is asking, can Dudleya survive wildfire?
Yeah, so that's a question. And hopefully that's somebody that's looking to do research and would like to make that their PhD study or master's or whatever it may be. So we don't entirely know, at least from what I read in the research in this. And so that's a big concern for like a species like verityi, and that's what we're gonna learn, how well they rebound from that fire. But typically from what I've read and from what it seems that it's most likely not the case. And maybe they've adapted in places where fires were just happened and limited their regions and made some rare species because of fire. But I don't know. And that's things that we need as scientists need to look at.
Tony is writing, is horticultural hybridization actually bad or detracting in relation to your expressed goals? Most horticulturally produced crops trend toward intentional selection, if not hybridization. If the idea is to grow plants for gardeners, why is hybridization a problem?
Hybridization is a problem for a couple of reasons. So if you were trying to conserve these plants in like almost like seed banking, and even for seed, and actually exactly that example, for seed banking, if you were to have a hybrid, then you're not conserving the species itself that you're targeting to save. So there's this happens with other species where it's called hybrid swamping, where you may have a more common species genetically flooding or swamping the genetics within a particular species of conservation need, or maybe a rare or an endangered species. A great example of this is out in Catalina Island with the Catalina Island mountain mahogany. But it occurs with Dudleya too. So if you are contracting or trying to grow, if a grower is trying to grow for conservation, then you need to make sure and take a lot of efforts to make sure that these plants are not near other Dudleyas and that they don't hybridize. And so that way you're able to conserve the genetics you're targeting to conserve. From a production standpoint, it's just, if I was a collector, which many of these poachers are and what they're trying to do, and if you're trying to, it's just trying to grow the plant that's desired. And so that's where doing clonal propagation, you're getting the species. You will definitely know that you're getting the species. And it brings up a good point about selection. And so there may be some species that are doing a lot that may be way more attractive or way more garden friendly. These are what the poachers were probably looking for, were some of these older specimens that are windblown and have just these awesome environments that where the environment over a lot of time has shaped these plants into these sculptures basically. And so if you're able to select for those type of traits and make plants that are more aesthetically pleasing than something that's in the wild, that's a great benefit to try to conserve these wild species by offering something in the horticultural trade that can then deflate that incentive to poach from the wild. And so I'm for hybrids in the horticultural trade. I think it can do a lot more good for these species if poaching is an issue as well.
Thank you. And well, I have a couple more. Well, let's see, 3 more questions written in. An anonymous writer says, I'm very interested in propagating Dudleya myself for conservation and decreasing the demand. What gear would I need to use? What's the best way to get started? And Vince wrote, where do we buy these kits? Do we need to start with a rosette or can we use any tissue at all? So I know this can be a fairly lengthy answer, but to condense it, what would you say to that?
Yeah, so traditionally, if you're trying to grow Dudleya, it's actually pretty easy to do from seed. If you get kind of viable seed within the first, I mean, I typically aim for the first year of seed production, but if it's properly stored, you can have that for a couple years. But sowing seed, you can use basically any traditional seed mix. A good one I like to use for the home grower is kind of a 2 parts perlite, or that, you know, that kind of volcanic sponge rock with a 1 part actual potting mix. So something you could buy from a nursery or garden center. For plant tissue culture kits themselves, the primary company I like to use, and that's kind of the leading industry for that, is Phytotechnologies. I can add these into the chat and things like that, but they are making it way more possible for people to purchase plant tissue culture kits and anything you need from plant tissue culture. I'm not sponsored by them at all. It's just a reliable source, and I'm really glad that company exists to make this more available to people. Resources. Okay, maybe that's another question that was coming in, but if there's a really good book for plant tissue culture, and unfortunately I have the old edition here, but it's called Plants from Test Tubes, and this is very well written. There's a new edition out there, but it has a lot of great information that's geared towards the home propagator and somebody who wants to do this and just getting into it. But also it's very well written and gives from anybody who's even wanting to start a business using this technology. So it's Plants from Test Tubes, and Kite and Clen, I think, is the authors for that. Again, we'll provide that information as well. Great.
And Kyle asked, is your work published anywhere? You would love to share it with colleagues.
I think it's been approved. I mean, I got my master's, but I think there's still a review process and things like that. It's a capstone project, so I didn't go in front of a review board itself for defense, but it's been reviewed and approved for, and so UCI, I'm not exactly sure how they're going about publishing that and making that available. But yeah, it's not published in any particular journal, but I think it's available at some point within UCI.
All right. Well, finally, and here's a good hypothetical question. David asks, do you have an opinion on a San Diego County Dudleya most endangered and that could most benefit from in vitro now?
San Diego itself, I don't exactly know. I think the attenuata orcudii is one that I think is in a region that's near the border, which there's a lot of challenges there that I think could definitely use any production as well. It's an excellent plant. I use it's really good if you saw that photo I had in my presentation, but it does really great for kind of rock gardens, and it's got a unique form. It's more compact. So, but that itself is another rare species. But I'm sure that it's really hard to say that because I know there's a lot more Dudleya especially out there that are in need help. So I just encourage that this is an additional tool for conservationists. I think I just want to make this available for more people and agencies so that they're not putting all their eggs in one basket, as I like to say.
All right, well, thank you again, Kevin. This was great. We really appreciate you speaking to us tonight. And that's going to be all for this evening. Check the chat right now for some of the resources. Our next presentation will be a collaboration with CNPS Orange County on Thursday, July 16th. And it's going to feature Professor and author Bob Allen and some of his students presenting the flora of Casper's Wilderness Park. And you'll find details on both chapters' websites and social media. And I want to thank everyone for joining us tonight, and we'll see you next time.
Thank you.
Bye-bye, everyone.